Ladies and gentlemen, distinguished guests, I welcome you to the first forum discussion at the ADPD meeting. May I introduce myself? First, my name is Manfred Windisch. I'm one of the co-organizers of ADPD conference since 10 years now. And I used to work in the field of drug development for neurodegeneration, but also cerebrovascular diseases for about 30 years now. I went through all the different developments of animal models. I developed and standardized some animal models myself. I was also in parallel always involved into clinical studies, so I know both worlds and I know about all of our failures. But before we are going in the middle of the topic, I want to explain you how this discussion will work. Please open your ADPD app. Go to the forum discussions at ADPD app and then click Ask the Speaker. This will allow you to write down questions and they will be immediately transferred to my iPad, and I can then address these questions to the colleagues here in the discussion panel. If your questions are general, I will ask who wants to answer, but if you want somebody to give you a specific answer, please indicate who should answer your question, to whom you're addressing your question. Is this clear for everybody? Has everybody downloaded the ADPD app? Hopefully, yes. We are sitting here today because this is now an experiment how well such a discussion will work. And we decided we don't do animal experiments, so I'm the guinea pig today. It's a human study to see how well it works and how lively you will discuss with us. You remember certainly we have now 20 years of intense drug development. We went through different targets. We did animal studies, but also clinical trials in thousands of patients. And as we all know, so far there is no approved drug available. We have the impressive failure rate of about 99.6%. And so we have to ask the question, what are we doing wrong? Is there any way to increase predictive validity of animal models? As you all know, we really rely on proof of concept studies in models where we believe that they are resembling at least key features or key pathologies of Alzheimer's disease, Parkinson's disease, or any other CNS disorder. Then we are extremely happy when we cure the mice. We are all enthusiastic. Maybe we read even something in the media about a spectacular new approach to treat AD. And a few years later, we read, unfortunately, this drug failed impressively in phase 3 clinical studies. I don't want to go into details what could be reasons of this failure. It could be more than the translational weakness of the models. And it seems that in particular, in CNS disorders, we are extremely vulnerable of this translational failure because it's not a problem of Alzheimer's disease. We have the similar findings in Parkinson's disease. We have a history of failures in stroke research, which was even that bad that at the end, drug development for stroke almost came to an end and hardly any studies are running anymore. It has an enormous economic impact because we all know there are millions of patients suffering from one of these diseases worldwide, and we are forced to develop efficient disease-modifying treatments. And I'm sure we will find a way out of it. And the start of all of these developments towards clinic is somewhere in the field of animal research. And I'm really happy that today I have 5 experts here who will share with us their experience, their particular point of view, how they see animal research, what shall we improve, can we improve something, what would be the ideal model. I hope we don't come to the conclusion that only the Alzheimer's disease patient is the ideal model. And to start with the discussion, and please, you can already think about questions you may have now. I simply want to introduce that all the speakers here in the podium are introducing themselves, and I am now handing over the word to Robert Cohen.
Hey, can you hear me? Turned on? So I've been working in Alzheimer's disease research since about 1981, primarily both in clinical trials as well as in basic research. And the primary reason I'm probably here today is because of a rat animal model that we developed starting at NIH, which is now available in the RRC for those people that are interested in using the animal model. I'm not going to go into detail about the model, but we had hoped that it might offer some advantages to the current mouse models.
I'm Frank LaFurla. I'm from UC Irvine. I've been working on Alzheimer's disease since 1990, specifically working on development of new animal models for Alzheimer's and related dementias. And I'm also the director of our Alzheimer's Disease Research Center, which involves analysis and clinical evaluation of 3 different patient subtypes.
And I'm Emer Leahy. I'm President and CEO of Psychogenics. Psychogenics is a preclinical CRO. We provide behavioral testing services, but we also provide a lot of non-behavioral capabilities as well, including electrophysiology, EEG, microdialysis, etc. But our core expertise is in behavior, and a number of years ago we developed some high-throughput behavioral testing platforms that combine robotics, computer vision, bioinformatics, and give us a very objective assessment of animal behavior. Using these models, we've profiled a number of Alzheimer models as well as other CNS disorder models, neuropsychiatric in general, and we have been able to detect early phenotypes and very robust phenotypes because of the massive amount of data that we collect on an individual animal.
My name is Mumin Pu. I'm director of the Institute of Neuroscience for Chinese Academy of Sciences in Shanghai. I myself is a neurophysiologist. I don't work directly on Alzheimer's or Parkinson's, but I have many colleagues who are interested in it. And recently, as one of the main organizers for the China Brain Project, in which Alzheimer, Parkinson's, and other neurodegenerative disease would be a main topic or main directions for the next 15 years in Chinese Brain Project. So I know something about the effort in this direction, and in particular, the use of primate as animal models for studying neurodegenerative disease. And I'll talk about that in a bit later.
My name is Joseph Ferruggio. I'm president and CEO of InterVivo Solutions, which is a contract research organization that focuses on in vivo animal testing. With respect to our mission, we're looking to develop more translational models, and within our repertoire for Alzheimer's disease, the model that's of most interest to us is an age dog model, which develops several features similar to Alzheimer's disease, including neuropathology, cognitive changes, and biomarker signatures.
So thank you very much for this introduction. As you can see now, we have people here from CRO industry. We have people from academia who developed some of these models, who standardized some of these models in CROs. Usually we are using validated and characterized models in a way to achieve highest reproducibility at least. And I think we are now ready for the first question. I see that there are already questions coming in from the audience. But first, just give me your personal opinion. What is the reason that so many drugs have been successful in the mouse models or rat models? I'm not sure how many have been tested in rat models and have been promoted to clinical research already. Some of the drugs replicated even a lot of the pathological changes which have been seen in the mice. So they reduced amyloid load, for example, also in clinical settings. They reduced amyloid in CSF. But finally, the weakest point, the weakest point in translation was always cognition and the validation of the cognitive improvement different between the models and the clinical trial. Do you have any opinion you can give about that, and how can we improve it?
Perhaps I can start. I think that the way we assess animal models needs to be very, very much standardized. There has to be adequate study design, which includes randomization, blinding of studies, and powering the studies. I think powering is incredibly important, and a priori statistical analyses plans going into the study up front to deal with missing data and to deal with outliers. With that, then, one can properly design cognitive studies. And I think that multiple endpoints need to be evaluated in animal models because we are dealing mostly with the familial models. So the models themselves may not be the most appropriate models for sporadic Alzheimer's disease, but given that a number of new modifiers are being identified almost on a daily basis, and with CRISPR technologies, et cetera, I think there's going to be additional models and perhaps even more useful models coming in the future. And that combined with good study design, I think we can address some of the cognitive endpoints.
So I would just add that I think we have to be careful how we refer to these animal models and what are they animal models of. The overwhelming majority that have been produced have just amyloid pathology, and I would argue that perhaps the overwhelming majority of clinical trial failures have occurred in animals that just have that pathology represented and not other key pathological features of the disease. There's no doubt about it in my mind that Alzheimer's disease is very different from what I would call Mouseheimer's disease, which is the development of the phenotype in these mouse models. Clearly, there's difference in terms of the expression levels. We know that there's also difference in terms of the amount of the protein that's overproduced. We know that there's a big difference in aging between the human and the rodent models, and I'm not so sure that age is as important as the aging process, which is very difficult, to reproduce. So I think we should be just very careful about how we refer to these animal models, particularly when we say that they're the reason that many of these clinical trials have not been successful.
I would add to that certain risk factors should be considered, and we know the key pathological events, but there could be other events that are involved in the early transition to Alzheimer's disease. So age has a risk factor. Multiple risk factors within an animal may provide some way to at least do later screening after target engagement models are run without a focus on a key pathology, and that might improve success as well.
I would like to comment on as a physiologist by training, I would like to comment on the pharmacological approach to neurodegenerative disease. The molecular targets may not be as the most appropriate target. One would like to have a neurocircuit target, a specific function that degenerates. The best example would be Parkinson's. Parkinson's, we know this dopamine circuit. And you have very good treatment for Parkinson, directly targeting, I'm referring to deep brain stimulation, direct targeting the reward circuit. And that's very effective. Right? So pharmacological target development has to be more specific to the circuit dysfunction. And for that, we need to have a better understanding of the circuit basis of degeneration. I mean, neurodegeneration, where does it start? Or where are the earliest stages we can target the early degenerating pathways versus general. So that would eliminate the problem of side effects and non-specificity effect.
I have another question here now. Sorry, I have another question because so far the majority of the substances for treatment in humans have initially been tested in transgenic mouse models. And there was a long debate about mouse and man. Now we have at least different approaches. One of the approaches is the use of rats. Maybe rats are already quite different from their behavioral spectrum compared to mice. Joseph is an expert in dog and cat studies, and Boo is working now with primates, and I think I heard that you are even interested in some transgenic primates. Is this right? And what can we now expect from new models? Will they create the same problem, or are they already one step closer to modeling the human disease?
I would just add that the mouse models have been helpful in the sense of being able to develop particular treatments for amyloid itself. So it's facilitated our clinical trials to try to see if eliminating or lowering amyloid in the brains would produce cognitive effects or benefits. And in that sense, it's been useful to say, you know, that just doesn't work, it's insufficient. And if you look at the animal models, at least in the rat, it's a very complex problem. Once amyloid is there, looking at messenger RNA, for example, in the rat model that I developed, we can see when we look at 15,000 basic sequences of messenger RNA, we see about 2,200 differences between the control animals, the litter-mate controls, and the animal that has amyloid present. And so a lot of these involve multiple systems, and this is after 2 months. So if you're looking at humans who have been exposed to amyloid for a period of time, we expect it to be a very complex problem that won't be treated just by eliminating amyloid or tau for that matter.
Any other opinion here?
I wonder if we need a cultural change if we're going to start moving away from mice to higher order animals, because there are clearly some significant issues when you move to these other species, not the least of which is that they're a lot more costly to be able to maintain. I mean, that's one of the advantages I think that we've had in the past about using mice, but correct me if I'm wrong, but I think it costs probably twice as much to house rats than it does to house mice. And at a time when the NIH budget is under very severe attack, I think we as a community of scientists are going to really have to lobby hard to make sure that those funds are going to be there if we're going to go to rats or to non-human primates and dogs. And thus far, we haven't done that or made that investment.
But can this be really just a matter of costs? Because if you calculate the economic impact of all the failed developments, the costs for more complex models might be neglectable.
No, it's not just cost, it's also timing as well, right? So we know that mice have a lifespan of about 2 years, and some of these non-human primates, are we willing to wait a decade or more for them to develop the kind of pathology? So it's a different type of cost, not just an economic cost, but also a time saving. And I would argue that we would have to come up with a different cultural model. Certainly, if you expect someone to get tenure at a university within 6 years, and you're using some of these non-mouse models, it's not going to be very practical. So it's just going to require that we change our approach overall.
Okay, I'm seeing the audience is extremely active, so my screen is filling up, and I think I have here a question which I had also on my list, and it's an interesting question because somebody asked, maybe we put too much emphasis on brain pathology and the reduction of pathology and not of the true biology of the proteins that are causing the disease. Maybe we have the wrong proteins. And I would add to this question, maybe we also did not pay too much attention on the behavioral consequences of the disease, because even in my experience, I had companies who said, okay, don't even look to behavior, that's not interesting. If we change amyloid, if we change tau phosphorylation, that's enough for us as a proof of concept.
Yeah, I call that the reductionist bias. I think it's very important to have a behavioral readout from a model in addition to other readouts, whether they're electrophysiological or biochemical. And one of the reasons that we developed the high throughput in vivo behavior-based platforms is to eliminate subjectivity and so that we can reliably measure behavior. And behavior resulting from some genetic manipulation in a mouse doesn't necessarily have face validity to what's happening in the human, but could very well still have predictive validity. So it's very important to get a broad picture of what's happening in the mouse and what's happening over time so that you can assess different treatments at different times during the course of disease. So what we have tried to do is, with robotics, computer vision, and informatics, really take a very broad picture of everything that an animal is doing, and in that way get a reliable and reproducible measure of behavior.
I would like to address the question of functional assessment of neurodegenerative disease early. Now, as we all know, the familiar curve of progression, the early signs is biochemical and the functional defects appear to be the latest. But I guess we need to develop sensitive functional tests. As an example, in Chinese Academy, we have a group of scientists who are monkey physiology, human psychophysicists. We, 10 laboratories got together to develop a broad spectrum, easy to use functional test kit for 10 different functions of brain. This is not disease oriented, this is a functional oriented. Every perceptual parameters, working memory, decision making, social recognition, all 10 different functions, you name it. 5 minutes each, you do a test on the brain, give quantitative assay of the functional state of the brain. And the hope is that we now have large populations line up for the regular populations, aging populations. We are monitoring them longitudinally for a long period of time and incorporating these functional tests into the regular annual health checkup. Now, we know we have all the annual checkup. We check everything in our body except our brain. So we need a quantitative assay with years, progression of our changes in a different function. And some of these functions will reflect, including movement disorders, it could be reflecting this function early. That can be monitored progressively early. And then, in the long run, pharmacological or physiological intervention could be introduced specifically aiming at those functions. And I think that might be approach for, in the long run, approach for early intervention, approach for preventing the progression of neurodegenerative disease before it's too late.
Thanks. I think I have to move a little bit. Into the questions coming from the audience, and I assume if somebody identified himself here on the screen, I can also name who was asking a question. I think here we have a very experienced researcher. It's John Hardy, who asks and who feels that mouse studies have been predictive in many outcomes except behavior. And he's addressing now a question if the new mouse models, the knock-in mouse models of Cyto, for example, are an improvement. And he also feels that many of the mouse studies so far have not been done in the right direction. So far I know John has also developed some knock-in mice or is in the middle of the characterization. What's your opinion about that?
So I do think that the Cyto model and the knock-in models are a significant improvement. I think a lot of people in the field, including my lab, are trying to use these knock-in approaches. Clearly, the significant advantage is that you don't have topic expression of these transgenes, nor do you have overexpression. You have physiological levels. With that comes a downside, of course, is that you're not driving the pathological phenotype as robustly. Now, whether or not that means it would be a quote-unquote less artificial, I think it remains to be seen. But certainly it seems as a field that there is a general shift towards using these knock-in approaches. We've taken the strategy that you've kind of alluded to, which is trying to use a knock-in approach to model sporadic Alzheimer's disease, because thus far the overwhelming, if not 100% of the models that exist for familial Alzheimer's disease, but yet most of the clinical trials are done on patients with sporadic Alzheimer's disease. So I think it is a trend that we're going to see. Again, I think it requires a significant cultural and financial investments, because it's going to take longer for these animals to develop pathology, and we're going to need larger numbers of these and more time.
So I'd like to add that a lot of the models are focused on a biochemical pathway or specific pathophysiology. And this is similar in several respects to some of the cholinergic models. And it just so happened that cholinergic drugs worked in Alzheimer's disease. And I think that might have less to do with the models than some of the other information. So clearly, if you knock down cholinergic transmission and then you supplement it, you can reverse those deficits. And I think we need to be cautionary when we're looking at specific pathways, because that would potentially be the same approach and really speaks more to target validation. One of the advantages of aged large animal models is that they demonstrate multiple characteristics and allow one to look at drugs in a black box system where we're not completely sure of what the pathophysiology is. But there's several features that map onto human Alzheimer's disease, ideally several biomarker signatures or biomarkers that can be monitored. So you can begin to look at human relevant changes in biomarkers, human relevant changes in behavior, including cognition and other non-cognitive type functions like stereotypy or behaviors that might be along the axis of psychiatric psychiatric effects in patients.
Okay, let's move forward, because now John Hardy asked this question about some of the studies have not been done very well. And another quite well-known person here in the room, Cristina Sampaio, asked me I should address the questions to the audience. What about reproducibility of results in these preclinical studies? What is the publication bias, because we do not know anything about negative data which have never been published. So how much impact would this have on translational failure, that we really have to assume that most of the studies have not been really conducted according to good scientific practice? Can you comment on this? What is your opinion? What's your experience about it if you read just the published data?
So I actually have some statistics on this. We spend on CNS research about 50 billion, and the conservative estimate of irreproducibility is about 50%, and it has been reported as high as 90%. So that is 28 billion conservatively that is as a direct result of irreproducibility. It comes down to, as I mentioned earlier, good study design and being able to report on negative findings, and also being assured that the positive findings that are reported in the literature are true positives. So is there an effort to reproduce those positive findings? Because otherwise, again, that will lead us down the wrong path.
Well, I would like to go back to the issue, which I think we were just touched upon, the use of non-rodent model. I would bring your attention to the fact that in Asian countries, starting from Japan's Mamasa project to now the China's Maquette project, on using non-human primates for generating neurodegenerative disease model. Now, the model is being generated now much more efficiently because of the appearance of CRISPR-Cas9 technology. We can introduce not only single gene, multiple gene knock-in or knockout efficiently in mice, in monkey. So I would say for in 5-10 years' time, we will have good AD models or PD models. And there is the problem of cost. Right? The cost is high, but there is devotion by the society in Asian countries to put resources into this area because this is really important for the world global health problem. Secondly, about the generation time. The monkey generation time for marmoset is about 2 years or 2.5 years. That's slow compared to mice. For macaque, it's 5 to 6 years for sexual maturation and getting second generation. But that has to be improved. Recently, we have developed technology to speed up sperm maturation by xenographing of the immature testis tissue in nude mice, and that speed up twofold. So now with hormone treatment, the generation time for macaque is now reduced to 2.5 years. So that's improvement. So now with even the solution of the generation of transgenic or genetically modified monkey, you can now produce in F1 population the first generation monkeys with potentially uniform genetic background, at least half uniform genetic background used by the use of haploid stem cell injection into the oocyte. So now the technology is moving at a very rapid pace. So for younger generations in the audience, in 5-10 years, you can go to a center in China or in Japan to do your experiments, just like the Parkinson model. Parkinson model for MPTP model is very widely used. It's not a good model, but the new genetic modified Parkinson disease model is being generated. That would allow you to do this disease progression more naturally as in human being. Right? So these are going to be available in the field within 5 to 10 years. And I think the hope that the primate model on the scene is real, is realistic.
Can I add to that? So I would argue that the reproducibility is an issue across all science, not just limited to neurodegenerative field or Alzheimer's. But that notwithstanding, there have been some studies that have been robustly demonstrated across multiple labs, across using multiple animal models, and perhaps there's no better example of that than immunotherapy against A-beta, but yet that still hasn't translated. So that's clearly not a reproducibility issue. Second point I would make is that, at least in my experience, and certainly when I give a lot of these talks, one of the things that I always show is that not every treatment that we have done in the lab has worked. And unfortunately, there's really no form for us to publish our negative results. But somehow that notion exists that everything cures mice of Alzheimer's disease. At least in our experience, that's not true.
Okay, I would like to address this question about the genetically modified primates. Do you think it's not only as difficult already from the costs to use such primates, and if you genetically modify them, are we not in the same situation as we are with the rodent models, that we have some genetic manipulations which we are usually not having in the clinical trials, because 99% of our patients are sporadic disease patients?
Yes, I think the genetic background is an issue. So the real to solve that issue, we need to clone the genetically modified monkey. I think the half clone using the haploid stem cell is on the way. That could be achieved within a few years. But the full clone of monkey is still a subject that has not been worked on very extensively. I know you probably heard about the Oregon Primate Center has tried it. They almost succeeded, but it was terminated. So to be a model, we need a large population of genetic uniform monkey, and the only solution is cloning. And whether that can be accomplished within the next few years is still not clear.
Can I just add to your question, Manfred? What I would be concerned about is licensing issues as well, because it's incredibly difficult to get a hold of certain animal models, mouse models, and so can you comment on the licensing?
Right. Not only the licensing, but also the social acceptance for the monkey research. I think we, in China, we are all in Japan. We have now faced on the situation that we, at the same time pushing for the primary research, we need to convince society. We have to do better work, better public dissemination of knowledge about the importance of a primary model in the long run for human health research. And this is, everybody agrees the importance, but the public hasn't been agreeable. And I think that a lot of work has to be done. Obviously, we need to set the highest standard, ethical standards to convince the society that we are doing this for the benefit of the human health. And I think that those are the work to be done. I think with the situation that face in Europe and the United States, in part, is because the society are not well educated in that sense.
You know, let's move from this discussion a little bit, because if we are already talking about doing experiments and drug testing in the primate models, in the audience I spotted a lot of interest in the question if we should use human cells, cells derived from stem cells, iPSCs, for drug development and drug studies. What's your opinion? What would be the best approach to have some, let me say, cell lines of humans which can be used for testing with a minimum of variability, would be fast, would not be animal test, and should provide close translational data at the end? Is this really true? Can we expect it? My personal opinion is the cells might be nice, might be close to humans, but they hardly behave, they hardly think, so can we overcome that problem? Do you have any ideas here?
I would add that it's almost impossible to imagine drug development without going into some animal species at some point. Regulatory agencies require animal testing for safety purposes. So the question I would send back is, why not do safety on people first? And if that's acceptable, then possibly, sure, we could move into neurons and then to people and skip animals. But not only do neurons not think, but drug absorption, drug metabolism is not ascertainable in that system.
I think it represents a new opportunity, and I think it may yield a lot of fruit for us. There's a lot of potential about using human iPS cells, in particular when you couple it with gene editing approaches like CRISPR, because now you could go in and take cells from an individual who is, let's say, harbors an E4 allele, and then just go in and modify it to a 3 allele without, changing any of the other genetic backgrounds. I think it offers a lot of potential. Whether or not they think or not, may be irrelevant if the underlying molecular processes are the same, but I think it's definitely worth an investment, and a lot of research groups around the world are working on this right now. Certainly at Irvine, we've invested very heavily in establishing an Alzheimer's iPS cell bank, and I think we have about 60 or so cell lines that we've derived from different patients, and we've already started to distribute that to researchers throughout the world. So I think it's going to offer a lot of potential, and we don't know yet whether or not it's going to bear fruit or not.
Forgive me if I'm jumping now a little bit, but there is obviously an enormous interest in the audience, and I have so many questions that we can sit here for the next 2 days, I'm afraid. And so I just want to pick a few questions which could help us for the next round of discussion. And there is something really for Joseph, he can hopefully answer it. Jianhao Zhang is asking, can you comment on the utility and concerns of aged dogs as a model? dogs of different breed have different lifespan and neurological phenotype as they age. So how are you addressing this?
Well, I think there's a lot of unanswered questions in dogs, and definitely breed differences are not well understood, and that's mainly for practical reasons. We use beagle dogs primarily. They tend to be the primary tox species, and so beagle dogs have been well characterized. In terms of utility, they're fairly easy to work with. The key events, including cognitive dysfunction and neuropathology, probably occur over a 4 to 5 year span between 6 and 11. So at this point, I don't think we can look at all dogs as a model, not because most dogs don't develop some of the pathology and the cognitive changes, but because we don't have enough information. And I think it's important to add that there's a secondary benefit of using dog. And so in the veterinary literature, there's a disease, and I'm sure if you have dogs, several of you will have noticed that when your dogs get old, their behavior changes. And this is referred to as cognitive dysfunction syndrome if it's not a medical condition. And the signs that you see in dogs are things like house soiling, not recognizing owners, changes in activity, increased anxiety. And so a lot of the work done in dog, I would argue, will hopefully translate to humans, and this is a big problem because we don't have drugs to test that. But there definitely would be a benefit for all of those breeds of dogs out there potentially. So at the very least, there's some economic savings in that if the dog is not the ideal model for humans, which is hopefully testable, it should be the ideal model for dogs.
Okay, thank you very much for this answer. I hope it was adequately answered the question. And I have now a question directed to Dr. Cohen from a person who is working with the rat model at the moment. And she wants to know from you why still most of the time mice are used instead of the rats when it turns out that the rats are the much better model. Are we all in agreement?
Well, I'd like to think so, but I think it really depends on what you want to create or have as a model. A model is not the same as human disease, and you don't want it to be the same. You want it to be simpler so that you can answer questions more rapidly. And so the question is, what are you using the model for? Is it understanding the illness? Is it to see what particular treatment affects a particular physiological system? These are the issues that have to be addressed. If we want to use something as complicated as the human, we might as well do the studies in the human. Maybe it's even more ethical than using a chimpanzee or some other non-human primate.
You know, there is now an interesting question coming in, and I had it also on my own list, and it also addresses a little bit what Dr. Pu told us. He said, okay, now an achievement should be cloned monkeys so that we have uniform animals. Usually in our mouse studies, we use inbred strains to minimize variability. But the question coming from Marco Prado addresses now the fact that the humans are genetically not homogeneous. They show a very wide variability, and therefore it's the question, what's the reason to believe that cloned identical models, inbred models are good models? Shouldn't we go into testing of outbred strains to have an increased variability, to get a more closer result compared to clinical studies where we are facing this variability as a reality? I think that's a question to all of you.
Let me begin with the monkey model for autism. Last year, my institute, two investigators generated a MECP2 overexpression case, used viral overexpression of human MECP2 in macaque monkey, produced in the first generation phenotype. And there are variable copies of human gene incorporated in different chromosomes. But the phenotype seems to be rather uniform, that they all show repetitive motions, stereotype locomotions, defects in social interaction among the group or in pairs for those transgenics. Also increased anxiety, some deficit in the cognitive function, but not severe, it's just the rate of learning is slower. All this, so the diverse genetic phenotype may not be a problem when you have overexpression of a particular gene, in this case MECP2. And the second generation we produced within 2.5 years also inherited, germline transmission inherited from the male donor, also show social interaction defects. So I think it's an issue. Genetic diversity, diverse background is an issue, but for some cases they may not be a big issue. Right.
You know, now I got a really interesting question, and I really hope that somebody here on the table, one of the experts, can answer it. Otherwise, we have to ask for help in the audience. The question was, what do you believe is the most important and critical difference between human brain versus mice or any other non-human? So I think that's one of the key questions we have, and I'm afraid if we could answer it, we would be much further in our drug development efforts. But are there any ideas in this round of experts?
I would say it's not just the anatomy and physiology of the brain, but it's the whole system in general, right? We know that the microme probably plays a role in Alzheimer's disease, as well as all of the other omes, you know, the epigenome, the connectome, the proteome, and so it's just an inherent problem we have when we go to, you know, an animal model. But I think it's something that we need to consider in general. And if I could just add on very quickly to the previous question, because I think it's somewhat related, not only is genetic diversity important, but also how we actually house our animals. Generally, we house them in a very deprived environment. And so that's also going to affect brain function and brain activity. You know, humans are not housed the way we house most of our animals.
Okay, you know, let's come back to this whole issue of behavioral testing, because in fact in the clinical trials we are always most interested in the functional outcome. So we use a different set of cognitive tests usually in all of these mouse models, nothing else. We are hardly addressing psychiatric symptoms and behavioral symptoms, which certainly are a big problem in the clinical setting. And on the other hand, in clinical trials, we usually use completely different tests compared to the animal tests. So for example, one of the standard tests is the Morris-Water maze about spatial orientation and spatial memory, and we don't have a similar test in the human field, for example. In Spitec, it would be very relevant because it really is one of the cognitive domains that is disturbed very early in the disease, and it's continuing, and also it is reacting to treatment. Have you any thoughts how we can improve the behavioral characterization of models?
Yeah, so this comes back to a point I raised earlier. It's really important to understand the complexity of the phenotype in its entirety. The hyperactivity, the aggression that you might see in your animal model, depression, anxiety, all of these features and how then they may translate. In cognition, certainly the Morris-Water maze is the favorite, but I think there are better tests of cognitive flexibility and executive function, such as attentional set shifting, which is equivalent to the Wisconsin card sorting test in humans. And there are even tests now of episodic memory in rodents, a sort of variant on the novel object recognition test. So I think there's a lot that we can do to improve on cognitive testing and other behavioral measures in mice.
And I would add, I think there's potentially a challenge as well, because clinically, I don't think some of the cognitive deficits and behavioral deficits are well defined early in the disease, which goes back to what Dr. Pu was speaking about earlier. And trying to have a clear understanding of what tasks and testing people to a greater extent earlier on, using that as a way to maybe map backwards to animals so that we have something that is translational in terms of tests?
Well, we all know there are a tremendous difference in cognition between man and mice. Now, I think there are conserved mechanisms for cognition. That's the cognition about the outside world, sensory perception, learning memory, even categorization, some form of decision making can be studied in mice. But we know in human or in primates, there are a higher level of cognition involving empathy, self-awareness, theory of mind, perhaps, or even human, the language ability, the complex vocal communication. So there are clear species difference, even for the depression. We don't really have a good model for depression. And I recall this amazing report from Isvan Modi's lab in UCLA. He talked about this, but I didn't see it in print, about this forced swimming test for depression. Now, one of his students had a mice shampooed to wash out the oil on the surface. The mice would float, would keep swimming, never get the state of floating on the water as a sign for depression. So the mice with shampoo never get depressed in this particular assay. So this suggests that our definition of animal behavior really has to be look at it in a very serious manner. And the parallel for human cognition, the conserved mechanism may be there, but the decision-making of mice is totally different from decision-making of a primate and a human. Now, the human decision-making takes cumulative evidence, an estimate of risk and benefit. And it's not a simple sensory motor transformation that one often associates with a decision in lower species or in rodents. So I think there are clear cognitive differences, and those cognitive differences has to be really recognized when we develop different animal models.
I think it's pretty clear that you cannot really compare behavior in a Drosophila with behavior in a mouse and behavior in a non-human primate and in humans at the end. I just want to leave this question out a little bit because there was another interesting question which is again very easy to answer because somebody really wants to know what is the best cellular target or pathway to prevent or stop AD. Because this could then be also the best to model, to do an animal model for the disease, and we would have a treatment target. Have you any ideas how to create animal models which are different from the animal models now? Because what we are doing, we are mainly modeling amyloid and tau pathology, and maybe we forget about other aspects of the disease.
So I would just say what we're trying to do is take a very long-term and systematic approach by first trying to develop a model of sporadic Alzheimer's disease. And the way we have done that is by assuming that the first requirement that you're going to have is to generate mice in which the human A-beta, APP gene is knocked in and that it does not harbor any mutations. As many of you know, that is a very slow and tedious process to do that, but I believe that model can then be used as a platform to introduce other GWAS-related genes and to humanize other genes. And it's going to be a very slow, tedious process, but I think we have to start down that road sooner rather than later.
So would this mean that we go now step by step and make knock-in models until we have a small human which still looks like a mouse?
Well, I think this highlights some definitional differences in the way people look at translational models. And I think there's a clear value in trying to understand mechanisms. But as a behavioral pharmacologist, when I think of translational models, the ideal, and this is purely within what I do, is to have an animal model where you test a drug and that outcome in that animal is exactly the same as what you see in the clinical population. And so I think if you're looking at translation from that perspective where you just want to screen drugs, then potentially it may be quicker to use aged animals, and maybe not just one species, maybe multiple species, so that you do cover maybe a wider range of age-related changes. But that approach might offer some benefit. The systematic approach, I think, will also is valuable, but will take a long time as well.
I think you're going to need multiple approaches. You don't want everyone doing the same exact thing, right? I mean, there's no point to that. We're going to have to have a lot of diversity in our approaches.
You know, Christina Sampaio again asked a very critical question, which is coming up also to my mind all the time. Her question is that tests in organs nowadays are fully accepted for toxicological investigations. So the most critical question would then be, do we need any animal testing for the drug development at all for the future?
As far as I'm aware, I think you still need to do in vivo animal safety and toxicity tests, at least in CNS. Maybe somebody else is aware of other information.
I think the animal models will still need to be developed for non-pharmacological tools, neuromodulation tools like DBS, like many of the non-invasive tools that be applied to different form of human brain disorders. For example, DBS application to depression, to drug addiction, to all this needs to be tested in animals to find a proper target. And a primary model for those diseases would be very useful before we go into direct human trial and error type of applications.
Yes, and as with the iPSCs, I think organoids, they don't reflect the brain, and you don't have the lymphatic system, you don't have the role of glia, so it becomes, it's still necessary in my opinion.
You know, I have the tendency to agree with you, of course, that we need the complexity of a living organ and living organism, an animal, for drug testing. And there is an interesting question here on my list. They are interested about all the comorbidities we see in Alzheimer's disease. The aged population doesn't only suffer from dementia, they have diabetes, they have hypertension, they have many other conditions, and could be the different outcome in the clinical studies have to do that these comorbidities are not existing in relatively young animals where we are testing, and how can we address this? Can we do some models of comorbidity? Does it make sense at all?
Yeah, I agree with that 100%. I mean, as the questioner asked, it's pretty rare to find the patient that just suffers from Alzheimer's disease. They usually have many other comorbidities. We've been trying to model that in our lab, and obviously that takes a lot of time and a lot of money to do those kind of experiments. And I hate to always come back to money, but it is a factor, particularly when you're dealing with age-related disorders. But we've looked at things like the impact of diabetes and hypoxia, ischemia, viral infections, and in general, they tend to exacerbate the underlying pathology, which I think is not necessarily a surprise. But I think it really reflects a challenge that we have in the field, even when we do, clinical trials, is separating out and stratifying the right patient populations that we enroll in these trials. And certainly, it's been the experience among the Alzheimer's disease centers that it's pretty rare when individuals come to autopsy to see them have only A beta and tau pathology. Most of the cases that we have, at least about 2/3 in the centers, tend to have a lot of mixed pathology in the brain. So you see a lot of Lewy body pathology, a lot of vascular pathology as well. So I think it's all related to the underlying question.
I think it's an interesting question. In aged animal models, comorbidities are almost impossible to eliminate. And that tends to be one of the major arguments people make against using aged animal models is you don't have a pure disease. So I think you can look at that from both sides.
And I think this comes back to also the question of inbred versus outbred strains. One can select certain inbred strains that have such comorbidities. I think the Fisher rat in particular is an interesting rat model for that reason, and perhaps why it shows such a robust phenotype, because it already has cardiovascular and other issues associated with it.
You can see, at least in the rat model, you can see, I think in some of the mouse models as well, you get significant cerebrovascular changes just due to amyloid itself. So you could imagine in the human, regardless of whether the person has hypertension or not, if they produce or have more amyloid present, that they were developing cerebrovascular problems, which leads to, under stressful conditions, even activation of region may produce inadequate oxygenation to that region, which leads to more pathology over time, which is why you almost always see vascular pathology, I think, in patients with Alzheimer's disease, part of the complexity of the disorder. Most studies in the mouse models, rat, often done at the youngest age where you see cognitive impairment present because it saves money and time to do that, but really it'd be better to do these in animals who are 20 months or 24 months old, would be more closer to the human in terms of comorbidities.
And shall we then also treat all the comorbidities in the rats or mice like we do it in the humans? Because it's certainly also a contributing factor to variability that they receive 7-8 different drugs at the end.
I think there's a fundamental problems with the comorbidities and overlap of symptoms in all the neurological and neuropsychiatric diseases. It's because our definition of the disease based on symptom is intrinsically not a good way of classifying the diseases. I mean, I think the Tom Insel has been arguing this for many years, and what I agree with him very, very much is that what we need to have the disease definition, especially psychiatric disease, is functional definition. Now, we don't define as a dementia or AD. We define it as a deficit in working memory, in episodic memory, in procedural memory. We do the definition of the function. And I long for days now, 20, 30 years from now, we have our DMS-10 is no longer a symptom-based diagnosis of diseases, but just a deficit of function with prescription of intervention and pharmacological treatment. And that's a way, in the end, to really solve these problems of comorbidity and this overlap of symptoms.
You know, I think there's another thing that's important to indicate as well, and that is that sometimes the humans are not particularly cooperative either. So my colleague, Claudia Kawas at Irvine, who actually just this week was announced as the winner of the Potamkin Prize, has been studying the largest population of individuals over 90 years of age. And one of her findings, which is very interesting, is that if you look at about half of the population of individuals over 90 years of age, they have enough pathology in the brain that a neuropathologist would say satisfies the requirement to meet threshold for Alzheimer's type dementia, but yet they're cognitively normal. They're not impaired. And then the other 50% seem to be impaired, but yet have no obvious pathology. So, you know, clearly there are a lot of multiple factors going in, even among the human population. So it's not just a problem with the animal models.
You know, that's a very interesting remark, and it goes along very well with the question which came from the audience. As you know, a couple of years ago, nobody was speaking about microbiota and microbiome, and now somebody here in the audience is really interested to hear if you think if we transfer human microbiota to the rodents, that we could potentially create better AD models. Do we have already some knowledge about the influence of microbiota on AD pathology and development of cognitive deficits and functional deficits?
I think it's a very important question. I would say that we know that in every single ecosystem that by far the most common organisms are microbes, whether they be bacteria, viruses, or fungi. And traditionally, the way we have studied them is to study them from a very isolated point of view. We study microorganisms in humans, or we study them in the environment. We very rarely combine them, and I think that's a new and burgeoning field that we're going to see develop. And I think it gets back to something that we often tell the undergraduates when we talk about the brain, and that sometimes we as neuroscientists forget that the brain is attached to the rest of the body, and that we do know that what you eat has an impact. We know that oral health seems to be very important for maybe underlying chronic inflammation, which can be a risk factor for Alzheimer's disease. So I know it's an area that's being hotly investigated right now.
No more remarks from here. I can see that the room is slowly emptying. Most likely there are many other attractive sessions here at AD/PD, what I certainly know and appreciate. So maybe we have a final round of statements. I would like to hear from all of you what would be, in your opinion, the next step to improve predictive validity of these animal models. Do we create new models? Do we refine models? Do we change the way how we test different drugs in the models? What do we do to increase reproducibility? And how can we increase the comparability to the clinical situation? Maybe everybody of you can give a few minutes of statement. Let's start with you, Joseph.
I think a lot of at least suggestions for those questions were raised earlier. So things like running studies under similar situations, reporting of negative data when studies are, let's say, properly run with the right subject numbers, right controls in place. And then looking at multiple models and comparing them across those standards. And I would add to that, it would be nice to use standardized drugs to potentially understand which models show at least some of the outcomes that we've seen in human clinical trials. So potentially looking at biomarker changes without changes in cognition, or biomarker changes with impairments in cognition, and beginning to map the model to the human outcome. And of course, that assumes, which for me is that translation means predicting a drug effect in a clinical population using animal models.
I would like to comment on the potential future for primate research in Asia. It's clear that this is very costly and it's big science. In the sense that a lot of the resources and personnel devotion has to be put into development of useful models. So I can think of the potential. In fact, the proposal is being done is under consideration for build an international center for primate research, where animal models are generated in a site in Shanghai, for example. And people can go there for visit, like you go to CERN for doing your accelerator experiments. You go there doing experiment, drug testing, whatever. The animal model facilities are all available. You perhaps have to pay a fee for the use of facility. And those are maintained at the highest quality for international use. And that could be a way for primate model, even also for marmosets in Japan, for example. Now you may have heard that all the disease, neurological and that has some genetic basis, are being produced in marmosets already by CRISPR-Cas and TALEN technology. They are waiting to see the phenotype. So in a few years, in 5, 10 years, there will be centers in Tokyo or in Shanghai where you can just go there to do your research. And those could be a future for the primary research that I can envision.
So reproducibility, there's so many facets to it. As Joseph said, study design is one, and upfront defining the studies, registering the studies, and being able to randomize, blind, and power up your studies appropriately is important. But also what's important is how you house your animals, all those little details. So what we've tried to do, and we've done this now with CHDI in Huntington's disease, is we've published on best practices for handling Huntington's disease mouse models so that the community can follow the guidelines that have been put out with CHDI. So that's one. I think we can also learn a lot from meta-analyses. It'll inform as to what would be the more reliable models that are available. And also with fill and trim, one can determine, whether the endpoint measures are reliable or not.
So many learned people say that this is the century of biology, and Steve Jobs has said that he thinks that the greatest advances in this century are going to come at the intersection between biology and technology. And I think as more and more of us have these wearable devices and we collect more data, and we didn't really get a chance to touch on big data, but as we accumulate more and more data, this represents an opportunity for manipulation in animal models. So I think the answer to all the questions that you asked earlier is yes, we need more animal models. We need to study them better. We're going to need more people involved with them.
So I'm a little contrary in the sense that I think we already have enough animal models per se. I think what happened is we got premature closure about what the pathways were to design clinical therapeutics. So as a result of that, almost all trials have been involved with reducing down amyloid levels, and we haven't looked at many of the other pathways that are probably involved in a disease. We already have models that are good enough, I think, to explore those, whether it be proteolytic pathways, messenger RNA pathways, mitochondrial pathways. They just haven't reached the point where people felt confident enough to try them in clinical trials. As the clinical trials have failed with some of these other agents, I think we'll be able to begin to explore and look at these other pathways for help for patients with Alzheimer's disease.
Well, thank you very much. Just at the end, there was an interesting question coming in that has to do with your statement now, Robert, because somebody is asking if we should not change the drug testing to other targets and other focus in Alzheimer's disease and define new biomarkers. I think that's certainly a very good question. It will need a lot of new development, and I'm sure that the research is ongoing and we will find some new targets, but what we have now is clearly models of aging, models of amyloidosis, and models of tauopathies, and some combined models. And I think first we have to refine them. We can maybe as a sideline develop some new other models, and it would be my opinion, now my personal opinion, maybe we move to a cascade of testing with drugs. First test them in organoids, test them in cell systems, pre-select, go into well-established mouse models, then move on to dog, higher species, and primate models, and only substances that qualify themselves to be moved forward in several of the models should then be promoted to the clinical. No idea if this is the right approach. It's certainly what I heard from all the people and the experts here in the panel. Whatever we are doing, the preclinical drug development and pharmacology will become more challenging and more expensive. So funding agencies should be aware that they provide more money for animal research that will finally pay off multiple times by improved drug development and successful drugs in the field. I think what I see now, we should come to an end with this session, in spite that there are still many questions, but we are turning back and back. My colleagues here, all the experts, have agreed to meet you here in this time we still have left over for a personal discussion if there's an interest. I really would like to thank all the people here on the stage for their lively discussion and their valuable contributions. And in particular, I want to address my gratitude to the audience. You have been great. You overwhelmed me with the questions. I think I still have to scroll through the questions for the next half hour. I think it was a real good success, and I wish you a lot of fun in Vienna, but also a lot of scientific experience, a lot of networking experience at ADPE conference. Thank you very much.